Hey everyone and welcome. Today we're diving into something truly mind-blowing, gene therapy. We're not just talking about treating diseases here. We're talking about a technology that literally aims to rewrite the code of life itself.
So, let's just start with a huge question. If you're born with a disease that's written into your very DNA, is that it? Is that just your fate? Or can we actually go in and correct that genetic mistake? Well, as you're about to find out, the answer is a pretty amazing yes.
And to see how, we've got to wind the clock back to a really pivotal year, 1990. This is the moment when all the theory, all the ideas became a reality. It's when the very first clinical gene therapy was actually performed on a person, and believe me, it changed everything. So what is gene therapy really? Well, the core idea is just brilliant. If a person has a gene that's faulty or just not working, scientists can introduce a correct, working copy to pick up the slack. You can kind of think of it like finding a typo in the source code of life and then uploading a patch to fix the bug. But this isn't just some abstract scientific idea. At the heart of this story is a real person, a little girl who was born without an immune system, and the incredible science that gave her a fighting chance.
The very first patient to ever receive this treatment was just 4 years old. She was suffering from a really rare genetic disorder known as adenosine deaminase deficiency, or ADD deficiency for short.
So ADA deficiency is a hereditary disease. It happens when the gene that's supposed to make an enzyme called adenosine deaminase is basically deleted or broken. And it turns out this one little enzyme is absolutely critical for our immune system to work at all. Without it, a truly devastating chain reaction kicks off inside the body. It all starts with that one bad gene. Because of it, the body can't produce the ADA enzyme. And without that enzyme, crucial immune cells called lymphocytes get destroyed. And if you lose your lymphocytes, your entire immune system just collapses. This condition is so serious, it's actually classified as a type of SCID. That stands for Severe Combined Immunodeficiency. You might have heard of it before as bubble boy disease, where patients are so vulnerable they have to be completely isolated from the outside world just to survive.
So, faced with this catastrophic genetic error, scientists came up with a procedure that was absolutely groundbreaking. This is where we really get to the part about editing the book of life.
The process they came up with was just ingenious. First, they took some of those key immune cells, the lymphocytes, right out of the patient's blood. Then, they grew those cells in a lab culture, a process called ex vivo, which literally just means outside the body. And this is the magic step. They inserted a working copy of the ADA gene into those lab-grown cells. Finally, they returned these newly upgraded genetically modified cells back to the patient.
You might be wondering, okay, how did they get the new gene into the cells? They used a super clever tool, a disabled virus called a retroviral vector. Seriously, think of it like a biological UPS truck. They basically took a virus, stripped out all the bad stuff that makes you sick, and loaded it up with the correct ADA gene. Then they used the virus's natural talent for getting inside cells to deliver that precious genetic cargo. And the result? It actually worked. The modified lymphocytes, now back inside the patient, started doing their job. They began producing the missing ADA enzyme, and her immune function started to improve. It was an absolutely massive success.
But, and in science, there's always almost a but. This incredible victory had a catch. It wasn't a permanent fix. And the reason why gets to the very nature of the cells they were working with.
So here's the crucial thing. Lymphocytes, just like most of the cells in our body, aren't immortal. They have a natural lifespan. They do their job, they get old, and then they die. And that meant the patient's newly corrected cells would also eventually disappear. She would need periodic infusions of the corrected cells just to keep her immune system functioning.
This challenge, this problem of the cure being temporary, immediately pointed scientists toward the next great frontier, the hunt for a permanent solution. The thinking was, if correcting adult cells is only temporary, what if you could make the fix at the very, very beginning of life?
And this slide really breaks down the difference. The 1990 therapy was somatic gene therapy. It targeted specific body cells, in this case lymphocytes, which, as we know, are mortal. But the potential future, the holy grail, is targeting cells at an early embryonic stage. Why? Because those early cells are the ancestors of all other cells. A correction made there would be copied into every single cell as the person grows, creating a permanent, lifelong cure.
Okay, that was a ton of amazing science to take in. So let's do a really quick recap of the most important takeaways from this whole genetic revolution.
All right, let's nail this down. First clinical gene therapy, 1990. What did it treat? ADA deficiency. The technique? That was ex vivo, targeting somatic cells, the lymphocytes. They used a retrovirus as the delivery vehicle. But the cure wasn't permanent because those lymphocytes just don't live forever. And the ultimate goal? A permanent cure, which might just be possible if we can do this at an early embryonic stage.
And that really leaves us with a powerful thought to end on. Gene therapy isn't just another medicine to manage symptoms. It's about going right to the source code of a disease and fixing it. It's not just treatment. It is, quite literally, correcting life at the genetic level. Thanks so much for joining me.